Toxoplasma gondii is a single-celled protozoan parasite in the kingdom Protista. It has a very complex life cycle with multiple hosts. Its definitive host is the intestine of cats. This is where feeding, growth, and sexual reproduction take place. Resistant oocytes are released from the cat's feces into the environment, where they are then accidentally consumed by one of a few intermediate hosts, particularly rats, mice, and birds. In the intermediate hosts, oocytes rupture, releasing the parasite into the intestine. The parasites then penetrate the lining of the intestine and spread through the body, forming tissue cysts, which remain dormant for a time. The tissue cysts are reintroduced into the feline definitive host when cats consume the tissue as part of their prey. So, when I started this research project, I was most interested in finding out how the Toxoplasma gondii parasite affects its definitive host, cats. With the parasites feeding and reproducing inside their intestines, one must assume that the effects on the felines cannot be insignificant. Yet, in reality, the Toxoplasma gondii parasite rarely causes significant effects in cats. This is part of what makes the protozoan parasite so effective. It does not cause enough harm to be evolutionarily beneficial for cats to develop mechanisms to avoid or neutralize it. Cats that have recently eaten infected meat usually begin excreting oocytes in their feces within 3 to 10 days and continue excreting oocytes for 10 to 14 days until their immune system stops the spread of the T. gondii. After that, they almost never restart oocyte excretion again. Although harmless in most cases, there are rare instances where a clinical disease known as toxoplasmosis develops. This is where the parasite buries deep into the cat's intestine and multiplies as a tachyzoite, because the cat's immune system is not strong enough to stop its spread. Toxoplasmosis is much more common in young kittens and cats with feline leukemia virus, or feline immunodeficiency virus, which is the cat's version of HIV. The most common signs of toxoplasmosis in cats are fever, diarrhea, vomiting, loss of appetite, incoordination, and lethargy. The parasite causes more specific symptoms depending on where in the body it spreads to. In the lungs, Toxoplasma gondii can cause pneumonia. In the liver, Toxoplasma gondii causes jaundice, a yellowing of the skin and mucous membranes. In the eyes, the parasite can cause inflammation of the iris and the anterior chamber, abnormal pupil size, and even blindness. In rare cases, a case of toxoplasmosis can result in death, especially when infecting the lungs or liver. A fresh T. gondii infection in a pregnant cat can also seriously endanger the fetus, resulting in stillbirths, abortions, and deformities. Diagnosing a toxoplasma infection in cats is not as simple as just examining feces for oocytes. They're not easily distinguishable from other parasites and are only released for a relatively short period of time. So, to determine if a cat has been infected, an antibody test must be carried out using their blood. High levels of IgG antibodies to T. gondii in a cat indicate that it has been previously infected, but is now immune and not actively excreting oocytes. High levels of IgM antibodies, meanwhile, suggest that an active infection in the body is taking place. The absence of any antibodies indicates that the cat has never been infected, and it could be in the future. Because T. gondii is a eukaryotic parasite rather than a bacteria, most antibiotics are ineffective in treating it. One antibiotic called clindamycin can be used, along with corticosteroids if there is significant inflammation. Treatment should be started immediately after diagnosis and continued for 2 to 4 weeks until signs have disappeared. No vaccines, however, are available to treat cats. There are also effective ways to prevent infection of cats. Felines contract T. gondii when they consume infected meat or unpasteurized milk. Thus, cats should only be fed commercially prepared cooked foods rather than raw meat or vegetables and pasteurized milk. The environment should also be kept free of rats that they might consume, or mice. Cats should not be allowed to hunt or roam about unsupervised, as outdoor cats and feral cats are much more likely to be infected than indoor cats. Cats should also be kept away from areas where livestock is raised or meat is packaged. But moving beyond its definitive host, cats, T. gondii is also important in the way that it affects its intermediate hosts. Although it is only able to reproduce within one genus, the feline, it has an extremely large range of intermediate hosts. T. gondii is capable of infecting any warm-blooded vertebrate, making it one of the most successful protozoan parasites on Earth. T. gondii can be found in everything from dogs, rabbits, sea otters, bears, deer, chicken, goats, pigs, sheep, and, even humans. A study of meat samples in the United Kingdom reported that 38 were infected with T. gondii. The protist's oocytes can retain their vitality for a long time, as can the cysts. For humans that have a healthy immune system, much like with cats, Toxoplasma cysts will likely remain inactive for life and not cause any issues. However, also like in cats, in immunocompromised people, such as AIDS victims, the Toxoplasma causes severe inflammation, as well as other issues. There is a further danger if a woman becomes infected for the first time during pregnancy. The parasite will spread through the placenta before it is contained, and the infection can cause severe damage to the fetus, which does not have a fully functioning immune system, resulting in birth defects. Most infected infants do not show symptoms at birth, but may develop debilitating symptoms later in life, such as loss of vision and hearing, mental retardation, and seizures. There are also some myths floating around about Toxoplasma gondii. In the past, Toxoplasma infection in humans had been linked with higher rates of schizophrenia, poor impulse control, aggression and violence, psychosis, and obsessive-compulsive disorder. Recent studies with better samples show that chronic infection of T. gondii in adults has little, if any, relation to such behavioral differences. It has also been suggested that cats should be kept away from pregnant women to avoid infecting them while their baby's immune system is still vulnerable. However, if you are a cat owner and are immunodeficient or planning to get pregnant, you don't really have that much to worry about. The reality is that humans becoming infected due to ingesting cat feces is pretty rare, and you are much more likely to contract Toxoplasma gondii from eating undercooked meat. Still, just to be safe, it is recommended that cat litter is changed daily. However, ultimately, while humans are an intermediate host for the protozoan, they're not a very effective one. Infection is much more incidental than intentional, as cats aren't likely to be, consuming human flesh. Thankfully, T. gondii has the most interesting effect on its intended intermediate hosts, those being rats and mice. Another thing I didn't mention yet as to why T. gondii is such a successful parasite is its ability to modify the behavior of its intermediate host. There are a handful of examples of parasites behaviorally modifying insect and crustacean host behavior. A famous example is zombie ants. You might have heard of these. A sac fungi called Ophiocordyceps unilateralis grows inside of ants' brains and controls them like a puppet to spread their spores more easily. However, parasite manipulation of hosts is very rare in mammals. The mammalian blood-brain barrier is normally totally effective in preventing pathogens from access to the central nervous system. But T. gondii can do it. After a rodent is infected with T. gondii oocytes, asexual reproduction begins rapidly, and cysts often penetrate into the brain within 2 weeks of infection. Unlike in humans, T. gondii does actually significantly alter the behavior of rats and mice to give it an evolutionary advantage by making it more likely for them to be eaten by cats. Thus, these cysts can be successfully reintroduced to their definitive host to produce oocytes. So, rodent behavior studies consistently show that toxoplasmosis is associated with impaired motor performance, deficits in spatial learning and memory, reduced anxiety, higher levels of activity, sensory attention deficits, slower reaction times, but most importantly, reduced avoidance of feline predators. Ordinarily, cat odors induce instinctive defensive behaviors in cats, making them extraordinarily cautious. However, rodents infected with the parasite Toxoplasma gondii are likely to not only lose their innate fear of cat odors, but to even become attracted to them. These infected rodents still continue to retain an aversion to the odors of other predators and do not have their sense of smell altered in any way, which shows that the altered behavior is not a mere byproduct of the infection. Instead, Toxoplasma gondii is precise enough to specifically alter rodent behavior in response to cat sense. Cysts that settle in rodent brains concentrate in limbic system regions related to both predator stimuli and sexual stimuli, which are important for innate approach and avoidance behaviors, and run parallel to each other in the limbic system. Studies find that in response to the scent of cat urine, T. gondii cysts will disrupt the defensive pathway in the infected rodent, while activating the sexual arousal pathway. Thus, in this manner, Toxoplasma actually makes rodents sexually attracted to cat odor, and thus much more likely to go near them and get eaten. A study by Patrick K. House in 2011 studied this phenomenon. You can see the results up on the screen, and they're a bit complicated, so I'll try to explain them to you. So in section A, you can see that rats infected with the Toxoplasma spent more time exploring cat urine than uninfected rats. In section B, you can see in uninfected male rats, female rat odor induced greater stimulation in limbic systems regions related to sexual arousal. In section C, it shows that in infected male rats, female rat odor and cat urine both induced around the same amount of sexual stimulation. Now, in section D, it shows in infected male rats, sexual stimulation was much greater when exposed to cat urine compared to non-infected rats. Section E shows that in infected male rats, female rat odor would still produce the same sexual stimulations as in non-infected rats. And section F, it shows concluding that Toxoplasma infection in rats clearly mimics neural activity in response to female rat odor with regard to cat odor. The mechanisms of how Toxoplasma cysts actually accomplish this altering of rodent behavior are still not well known and a significant subject of interest for scientists and researchers. However, it is assumed that the phenomenon is related to the neurotransmitter dopamine, which is related to decision-making and reward. Some studies have found that whole brain dopamine levels in infected mice are up to 15% higher than in non-infected mice. The Toxoplasma genome contains a homologous of tyrosine hydroxylase, which is the rate-limiting enzyme used in the vertebrate biosynthesis of dopamine. Thus, it is entirely possible that Toxoplasma parasites are increasing dopamine levels in infected rats by synthesizing the enzyme tyrosine hydroxylase. Increased dopamine levels would also explain why infected rodents are more hyperactive. Cats are attracted to moving and exposed prey, much more so than stationary or hidden ones. As stated before, common symptoms of toxoplasmosis in rodents also include generally impaired motor function, deficits in spatial learning and memory, sensory attention deficits, and slower reaction times. These all make rodents much more likely to be eaten by cats. However, it should be noted that these generalized effects only appear in mice and not rats, which are much more resistant to the parasite. Mice who suffer from a chronic infection have been found to have a much higher concentration of T. gondii cysts in their brain compared to rats. For example, a 2011 study by Miroslava Baranretrova, sorry, that name's very difficult to pronounce, found that the absolute density of T. gondii cysts in rat amygdalas was on average 0.15 cysts per millimeters cubed, compared to 2.1 cysts per millimeters cubed for mice, which is a factor of over 10 times more. Despite infected rats becoming attracted to cat scent, they are generally healthy and behave normally otherwise. Infected adult mice, meanwhile, show signs of acute infection and can suffer from toxoplasmosis-related inflammation of the eye, inflammatory lesions of other areas, enteritis, pneumonia, encephalitis, and myocarditis, which may cause death. Studies using laboratory mice inoculated with T. gondii show clear diminished learning capacity and memory in double-training maze experiments compared with non-infected control groups, sometimes running in circles and bending their heads to the side. Scientists hypothesize that these effects in mice are caused by local nerve degeneration around chronic infection. The T. gondii parasites might directly cause the death of infected neurons or disrupt their normal processes, or cause swelling and inflammation. Magnetic resonance imaging studies in chronically infected mice shows dilated ventricles and significant loss of brain tissue, and the cerebral cortical areas of the brain responsible for coordinating movements almost always had higher concentrations of T. gondii cysts than subcortical regions of the brain. These findings seem to support this hypothesis. So, in concluding this seminar, I wanted to end with an interesting thought that I came up with. You see, while Toxoplasma gondii is often referred to as a parasitic organism, and I just referred to it as a parasitic organism throughout this seminar, I don't think that that is always accurate. Parasitic implies one organism is being harmed for the benefit of another, but most cats are never harmed by T. gondii. T. gondii can be argued to have a commensal relationship with its host, where one organism benefits and the other is unaffected, but that would only apply for domesticated house cats. For feral and free-roaming felines that need to hunt for their food, I would actually argue that T. gondii has a mutualistic relationship with its host, where both organisms benefit from each other. T. gondii uses cat intestines to feed and sexually reproduce, and returns the favor by infecting a cat's prey, altering their behavior, and making it easier for them to hunt and obtain food. And rats especially are one of the most innately cautious mammals known, so the help that T. gondii provides its feline predator in catching them cannot be undersold. So really, it seems to me that cats and Toxoplasma gondii have one of the most legendary partnerships in nature.